All right, so the next topic is membrane transport. We'll see how far we get before this video gets too long. So the first thing you need to know is the two basic categories of transport. You have passive transport and active transport. If it is passive, it means that no energy is required. And anytime we say energy, we mean ATP. It is the universal energy currency used in your cells, or almost universal. And so if it's active, it requires ATP or energy. We are going to start by talking about the passive processes, those processes that don't require ATP for them to take place. And we will cover them in this order. So first up is simple diffusion. This is when something that can pass through the phospholipid bilayer passes right through the phospholipid bilayer with no other kinds of molecules getting involved. That's why it's called simple. All of the forms of passive transport act on the same principles of diffusion, wherein molecules will move from an area of high concentration to an area of low concentration. This happens anytime the temperature is above 0 degrees Kelvin. Which is absolute zero or negative 200 and something degrees Celsius. So as long as there's some sort of ambient heat, molecules are moving around. When they move, they bump into each other. When they bump into each other, they spread out. So molecules want to be as spread out as possible. If you have an area where there is a high concentration of molecules, they will be bumping into each other and automatically will try and move to an area where there is a low concentration. This is called moving down the concentration gradient, just like a ball rolling down a hill. And the big example that you always give if you're standing at the front of a classroom, you spray some cologne or perfume in the front of the room. There's a lot of cologne molecules up there, but then over time they'll make their way to the back of the room and people will be able to smell the cologne in the back of the room as well as the front. So channels are one way for nonpolar molecules to move into or out of the cell, but they're still going to be going down their concentration gradient. So here is an example of a protein channel. They tend to be specific for what moves through them. So some channels let sodium through, other channels might only let potassium through. And whatever they're letting through is always still just moving down its concentration gradient. We will talk about the other specific types of channels when we get into more of the electrophysiology of membranes. So just for now, know that channels are ways for small dissolved molecules to pass into or out of the cell down their concentration gradient. Carrier proteins are similar to channels, except they tend to operate on bigger molecules. So if you had a protein with a hole in it big enough for glucose to pass through, lots of other things would pass through it. So when you have bigger molecules like glucose or amino acids, you have to have what is called a carrier protein that, if you look down here, it's always sealed on one side of the protein. So over here on this side, they're showing you lots of glucose outside the cell, not so much glucose inside the cell. So the glucose molecules bump into each other and then shove one another into this carrier protein that then dumps the glucose inside the cell. Carriers work in both directions, and they just work, again, with the concentration gradient. So it's a way for larger molecules that can't pass through a channel to go from an area of high concentration to an area of low concentration. Osmosis, we are probably going to talk about in a little bit more detail or a little bit slower in class. This is a special form of diffusion wherein water is diffusing through the cell membrane. And it doesn't matter whether the water is diffusing through a protein pore, which is called an aquaporin, or whether or not the water is passing through the phospholipid bilayer. And it's important to remember that water can pass through the phospholipid bilayer. So the cells can't really control which way the water wants to go. We are going to skip ahead down here. So you have two aqueous solutions on either side of a membrane that is permeable to water, but not to the solutes in it. So think a sugar solution that can't cross the cell membrane. You have on the right here high solute concentration, on the left low solute concentration. What you need to remember is that the solute takes up space. So if you have more sugar over here in the same volume of water than you have over here, there's more water on this side, less water on that side. So wherever there's less solute, sugar, salt, amino acids, whatever it is, wherever there's less of it, there's more water, and water just travels down its concentration gradient from where there was lots of water to where there's less water. So it's really overall pretty simple. It's just water diffusing the same way everything else does. It's just weird to think about how much water is in a given volume. It does have its own concentration. So yeah, when osmosis occurs, water enters or leaves cells. And let me adjust one thing here. Sorry, there we go. So this then leads to three different terms that we need to define. The first is isotonic. So if you say a solution is isotonic, then it has the same solute concentration as the cell. This would be like a saline solution. If you've ever had a cut and they say they're going to clean it out with saline, it's a solution that has the same concentration of salt as you have stuff inside of a cell. So in a condition where the solution is isotonic to the cytoplasm, there's no net movement of water. Water will move into the cell and out of the cell at the same rate. If you have a hypertonic solution, hyper means more or higher than. So there's a greater concentration of solute in the solution than there is of solutes in the cell. So there's more water in the cell, less water outside the cell, because outside the cell is all salt and stuff. This then causes water to diffuse from where there's lots of it inside the cell to where there's less of it outside the cell, and the cell shrinks up. Hypotonic refers to a solution that has a lower solute concentration than the cytosol. So think distilled water. It's just water with no solutes in it. So when you put a cell in a hypotonic solution, the water goes from where there is lots of water, the solution, into the cell, and the cell swells up. So that is a quick overview of passive transport, ending with osmosis. And we'll stop there.